So welcome back after lunch. Our next talk will be data visualization for the web with Altair and Vega-Lite by Patrick Muehlbauer.
Okay. Yeah, hi. Hi. Thanks for having me here. Thanks to the organizers for making this conference happen. I'm Patrick. I'm a software engineer working for Yonder, and I'm mostly a backend engineer, but nowadays I'm working on our front-end application and work closely together with our data scientists and data engineers to bring visualizations to our front end so that our customers get better insight in what we are actually doing. And what we are doing, you heard today in the keynote by Felix, so I give you a short version. We predict the demand of various products, mostly bananas. And so when you go into a grocery store and want to buy a banana, so that there actually is a banana to buy and it doesn't look like this. On the other hand, we also try not to produce any waste, so we cannot just say, okay, order 1,000 bananas and that's it. When our data scientists do their calculations, you end up with data that roughly looks like this. So here's the raw data. It's just a table where you have for all day, product, and store combinations what the sales were that's sent by our customers and what we predicted. Usually there's lots of more columns with other data like stocks and so on. But yeah, so and if you have only data in that format, it's really hard for us humans to see what's going on. So what we usually do is we create visualizations like this here, where we just have a curve for our sales and a curve for our predictions, and can see pretty fast what's going on. And yeah, so we don't only want those visualizations for us. So not only our data scientists want that, but we also want also our customers want to have such visualizations. So we kind of need a way to make the plots our data scientists create in their daily work, make them available for our customers as well. So, and Python has a really big landscape of visualization libraries. There's things like matplotlib, you have Seaborn, pandas plotting, and but not all are that well suited to integrate it into web applications. So what we tried first, okay, we had that, was Bokeh. Bokeh is a really nice visualization library and already targets modern web browsers. So that seemed like a really good fit. We already use Python, and in Bokeh you also define everything as Python objects and so on. So you can get started really fast. The problem kind of was that Bokeh generates all the JavaScript for you, which can be nice, but for our front-end developers it was actually more of a burden, because our front-end developers don't work with Python. They use technologies like JavaScript and CSS, and they want to create their visualizations also in that environment with libraries like D3.js or highcharts or whatever. And the problem with Bokeh.js kind of was that it came with its own dependencies, and sometimes the dependencies of the front end we already had and the dependencies from Bokeh.js clashed. So there was, for example, some library in version X required by Bokeh.js, but in our front end there was already the same library in another version. And yeah, another problem is if you embed those Bokeh charts in your web application, it's not that easy to make the whole application look like one. And because when you create UIs for your customers, you want that it looks has the corporate identity and such things. Okay, so what we looked at then was Vega. Vega is a visualization grammar, and you define the design of your visualizations in a declarative manner. So you create JSON specifications, and those JSON specifications can then be used by any renderer that understands those specifications and in any environment. So for example, if you write a renderer to render such a specification in a web browser, then you can just render the graph in a web browser. You can do the same for any desktop application. Yeah. And Vega is kind of low level. It's more like a base for a whole ecosystem. What we mostly use at the moment are Vega-Lite specifications. Vega-Lite specifications are a more concise form of Vega specifications, and those specifications compile to Vega specifications. So here, this JSON is, for example, a bar plot for a Vega chart. So we kind of have to use it in Python and make it available to our front-end engineers. So what you could do is create those specifications by hand, expose it via an API endpoint, and the front-end developers can then just consume this service and render this JSON specification or do whatever they want with that. But creating those specifications by hand would of course be pretty cumbersome. So that's where Altair comes in. And with Altair you can create such Vega or Vega-Lite specifications via Python. Like you can see here, the data is defined via pandas data frames. I think we all know pandas. And the values that are here put into this data frame are actually the same like here. So that's translated one-to-one. And you can get the same Vega specification. Okay, so now I have a demo. I think I will sit down for a moment for that. Like I already said, you only need a renderer to render those specifications, and Jupyter Lab by now provides such a renderer. So you can just use Altair directly in Jupyter Lab notebooks, and the charts are shown automatically. And that's available by this import here. Import Altair as alt, and yeah, we can start. And as an example data set, I have here, I scraped Twitter. I have all the profiles, all the followers of the EuroSciPy Twitter account from Sunday, so maybe not everyone is in there right now. And it looks basically like this here. We have all the accounts. We can see the name of the people, how many followers they have, and so on. So, and if we want to create, for example, a bar chart out of that with Altair, we can put in this data frame into the Altair chart class. With those mark underscore methods, we say how the data should be visualized. So here we say we want to have bars. And in this encode method, we map our data to visual properties. So we say the screen name, which is the Twitter handle, should be visualized on the x-axis, and the number of followers should be the y-axis. And that's all to specify our first bar chart. We can actually encode more info than just x and y. We can also encode color or shape. So when we say we want that we want to have color for each Twitter handle, yeah, we get colored bars, and we even get a legend with all the names. That's not really useful but it's just as an example. Okay, so we can do more than just bars. We can also do scatter plots, for example. And I reduced the data set here so that we can actually get nicer plots. So if we want to create a scatter plot where we say the y-axis should be the number of followers and the x-axis the number of tweets, we say, okay, we want it visualized as points. The rest is like before. And we get a scatter plot. There are lots of different marks. Instead of points, we can also do circles. Now with the number of accounts we actually follow here on the x-axis. And yeah, we get another scatter plot. I'm not sure why mark circle produces points and mark point circles, but yeah. We can do the same with squares, and we can even add more properties to our marks, so we can make them transparent if we want. So to make our charts a little bit more useful, so up to now we don't know which points those here are, and we can add tooltips. That's as easy as saying, okay, we want a tooltip and we want to show our screen name, the Twitter handle, and the number of followers. And now we can just say, okay, here, that's David Martin. Maybe he's here, I don't know. So Vega charts can also be interactive. Interactivity with Altair is achieved by calling the interactive method on a chart object. So we can make our axes a little bit interactive. We can zoom, we can drag them around. Yeah. What we can also do is we can compose charts. We can use the charts from before and concatenate them. So here we say we want horizontal and vertical concatenation. That's done via altair.hconcat or vconcat, and there's even a short form. There are operators, so just or and. And now we can actually have all those 4 plots in one. To make that actually useful, we can use selections. So selections are the building blocks to have interactions in Altair. So here we define a linked brush. It's a selection where we say, okay, we want an interval. So if we mark certain points in a plot, then they are highlighted, for example. So we will do that here. We create the same chart as before. Actually, as a base chart, because we only say what the y-axis should be. We define x-axis later. And our color will be a condition with this brush. So what we say here, if a point is in the selected area, it should be blue. Otherwise, it should be gray. And we add this brush with the add selection method. And yeah, here again, we concatenate all those charts, and how you can see here, we can even define what the x-axis should be later on. And that's what we do here. And now if we mark points, we can see in the other charts which points we marked here. Okay, so there are various other selections. For example, we can also bind the scales to this brush. So what we have exactly the same. The only new thing is this bind equal scales. And now we again have this zooming. And if we zoom here and points are not visualized in that chart anymore, those get gray in the other ones. Crap. Okay, so those just as a few examples of what you can do with Altair. What's funny or interesting, you can even create Altair chart objects by parsing Vega-Lite or Vega specifications. So we use one of the charts we had before, say we want to get the JSON representation of that, and then we parse it again, and we get the same chart like before. Okay. So, okay, to summarize, so Altair provides us a declarative Python API to create Vega and Vega-Lite specifications, so we don't have to do it by hand. And we can use all the features of pandas to specify our data. Altair has this concept of encodings we have seen, so that's how we map the properties of our data to visual properties like color, shape, and so on. We can define different marks like bars or points, etc. Selections can be used to create interactions. And Altair doesn't care for how the graph is visualized or what is responsible for rendering. So that's done by other things. So for example, Jupyter Lab ships the renderer and not Altair. Okay, so but what we actually want to achieve is now that we can use the same charts in our web application. So we have to embed it in our HTML, for example. And that's done with the Vega embed JavaScript library. Here we have a really short JavaScript code. That's our specification. That's just a dictionary. We have some options, and then we use the Vega embed function to say render this back in this container here, which is just this div element. And how this can look like in a real application, I have here as a demo. So what we have here is a simple web formula. We can select a date range. We can select a store. So let's select Trento. And we can select different products. So let's take our banana. And then we can execute this formula. And we get a graph. So what happens here is when we click render plot, we send the selected data here to a backend API and get back this a Vega-Lite specification, so just a JSON, and then we render that JSON here. Okay. So the code that is needed to create this application. So in our backend, we have two endpoints where we just return what stores are actually available and what products. So that's what's used for this autocomplete feature. And So I used Flask for that, and what I did is I just loaded the data on service start and added it to my application object, and that's just a data frame. And then I return basically a list of records, so with product ID and product name, or store name and store ID. The actual function to get the Vega specification can look like this. So we pass our query arguments, the product, the selected store, and the date range. And then we use pandas query functions to select only that part of the data frame we need and create our plot. And to create the plot, we have to transform the data before. So I showed you the table we had with sales and predictions as separate columns, but so that's a so-called wide form, but Altair better works with long form. So what we do a meld. We say transform the data frame in a way that the sales column and the predictions column get actually one column. That it will call it legend, and the index will be pieces, so that we can still differentiate what is a sales value and what is a prediction value. And then we can just put that data frame into an Altair chart class. Say we want a line chart here, and yeah, that's it. What I haven't mentioned yet is this. So with this :n, we say what kind of data that is actually. So that would be nominal data. I haven't used that before because when you use pandas, and then Altair can actually infer the type from the pandas data frame. So it then knows, okay, that's an integer, so I have to use that data type. But it would actually be good practice if you always specify what kind of data you want. So there's also ordinal and quantitative data and so on. Okay. So in our front end, I used Vue.js and Beautify.js here, but you can actually do that with any framework or plain HTML and JavaScript. But with Beautify, we get this autocomplete widget for free. So yeah, what you have seen is just this v-autocomplete. We put our products data in it, and it just works basically. And we have a simple button and our container where we want to render the plot. The code will be available on GitHub later, by the way, so I will tweet where you can find it. So in our JavaScript code, yeah, that's Vue. So here this created function basically says when we load the web page, then we want to execute, this will be executed. Here we want to execute this fetch stores and fetch products functions, which just fetches the list from the backend we have seen before with all the products and stores available. And when we click the button, we want to we call that function plot data, where we send the selected store, product, and date range to our backend. And then we use again Vega embed to render the actual chart. Okay, so and yeah, you have seen that before. So to summarize, Vega is yeah, a grammar which can be used as base for creating visualizations. So it's pretty nice because yeah, you actually don't have to care about how it's visualized. You just have to define what do you want to have. With Altair, you can create those specifications via Python. And Yeah, with such Vega specifications, it's really easy to render those charts in any environment. So we have seen that it can be rendered in JupyterLab, in web browsers, but yeah, maybe it would be even possible to write a renderer for a terminal. I don't know. Okay, so that's it. Yeah, we are hiring. So if you are interested to work on data science problems, data visualizations, you can come to us. Yeah, I'm done.
Questions?
Thank you for your talk. A quick question about roughly comparing with Plotly.js. This is the framework I'm using now to render plots, and it's kind of highly customizable. How the Vega, not the Vega because this is a specification, but how far you can go in customization of your graphs, you know, like adding annotations, you know, custom legends and stuff like this, compared to, I mean, quick comparison. I mean, because maybe I'm going to switch to this one because it seems to be really good.
Yeah, and in comparison to what?
Plotly, I don't.
I actually don't.
Oh,
so
you don't use
I
it?
know that there is Plotly, but I never used it, so.
Okay.
So I
Okay.
don't know.
Okay. Thank you.
Any more questions? So it seems there are no questions. Thank you. Next talk will be When Less is More, Dimensionality Reduction in Neuroscience by Pietro Marchesi.
Scheiße. Hätten wir einfach mitnehmen sollen. Oh, ich habe das mitgeklaut.
Okay. How does this work? Hi, everyone. Okay, so my name is Pietro Marchesi, and I will tell you a little bit about dimensionality reduction and why it's interesting for us in neuroscience to do it. So, quick show of hands, who has never heard of dimensionality reduction? Okay, very few people. Who has worked with electrophysiological recordings in? Okay, also a few people. I hope I don't bore you. I am trying to step back for everyone else. About me, I work as a neurodata analyst for the Cognitive and Systems Neuroscience group of the University of Amsterdam, which is led by Cyril Pennertz. And broadly, we're interested in mechanisms by which the brain combines information that pertains to different sensory modalities. So we constantly are exposed to stimuli in different senses, and our brain is always trying to combine those and allow us to make sense of the world. And the way we research it typically is that we have animals, in our case rodents, who are engaged in tasks of different types. And as these animals are performing these tasks, we go and record the activity of individual neurons, populations of individual neurons in different areas that we are interested in. And when I say we, I mean my very talented colleagues. I am trained in physics, and I only get to play with the amazing data that they collect. I don't actually do the hard work. But in any case, once we have this data, the main challenge is to understand the mapping between what the neurons are doing in the brain and the sensory input that the animal is receiving and the behavioral output that it is generating. And you may rightfully wonder, why is this difficult? And I can think of a few reasons. So for one, neurons have typically complex responses. So there are neurons that do simple things, but normally they respond to a variety of the task parameters that we have in our experiment, a phenomenon known as mixed selectivity, which of course makes it difficult to understand at the single neuron level what exactly is this neuron doing, what is it computing in the brain. Also, neural responses are very variable in the sense that if we present an animal with the same stimulus twice in a row, we'll get different responses. Also, we record many neurons at the same time, and so when we look at this population, it's hard to kind of grasp what is the kind of this population computing, because we have many neurons that we have to consider at the same time. And lastly, also the behavior that these animals are doing can be complex and variable. So that's an additional layer of complexity. And so in this setting where kind of the data is very complex and it's not always easy to make sense of it, dimensionality reduction comes in as a way that can allow us to make our data, if not simple, at least a bit simpler. And the intuition is that we have a neural process which is underlying behavior, and we can explain that process with a few latent variables which capture a lot of variance in the neural activity, and hopefully these latent variables are going to be fewer than the number of neurons that we originally recorded. So the idea is that we can go from a data set where we have many neurons recorded over time, apply dimensionality reduction, and we get a few, hopefully intelligible and interpretable latent variables with which we can make sense of our data. And I put here also two almost universal facts. Number one is that the dimensionality of neural data turns out to be very often much lower than the number of neurons that we record, so the system is quite redundant, if you will. And also the low dimensional representations that we get are often pretty nice, in the sense that they make quite a bit of sense generally. So again, to further illustrate the idea of doing dimensionality reduction on neural data, suppose we record the activity of 3 neurons over time. At every point in time, we get a point in this 3-dimensional neural space, which is the activity of these 3 neurons at a given point in time, right? And you can see, aside, you can stop me anytime and ask questions if you have them, so don't be shy. So we have this trajectory through neural space. But then it's not hard to imagine. So can this trajectory explore every corner of neural space? The answer is probably no to that question, and the reason is neurons are embedded and connected in a network. So probably there are constraints related to their connectivity that prevent them from going exactly anywhere they want in neural space. And in the simplest scenario, you can imagine that, for example, this trajectory lies on a 2-dimensional plane embedded in this higher dimensional space. And so we can identify the latent variables that span this high dimensional space, and this allows us to go from a high dimensional description where we have all these neurons to a lower dimensional description with only these latent variables which capture patterns of coordinated activity expressed by these neurons. And there's something quite specific, I think, about doing dimensionality in neuroscience. So, yes? Yeah. Yeah, so it depends a little bit. Normally it would be, for example, a firing rate. So we record discrete events. So we record the action potentials of the neurons, and then we smooth them. We get some continuous measure of their firing rate. So that's what it would be. In other cases, it would be calcium expression or something similar. Yeah, so thank you. Good question. So why dimensionality reduction? The first reason is very obvious. It's good to represent your data efficiently and parsimoniously. So if we can get a good representation with fewer dimension, that's always great. But there's a second point, which I think is quite specific to neuroscience, And it's the idea that through dimensionality reduction, we can actually unveil mechanisms that are occurring at the level of the population that we otherwise couldn't really extract from looking at the single neurons. And this goes hand in hand with the idea that if we can identify the latent variables that span these low-dimensional manifolds on which neural activity is constrained, then these latent variables are actually the interesting thing that we should be looking at. So instead of the single neurons, which are canonically the object of interest of the neuroscientist who analyzes data, maybe these latent variables which span the manifolds, also called neural modes sometimes, these maybe are the interesting things. And I give you one example of this because I think this can be a bit difficult to understand. So in this little example here, there's a simple task in which essentially, very simply put, a monkey gets a stimulus, has to remember it for a few seconds before it can use that information to do something, right? So this is a working memory task. And if you look at the single neurons, there's a few neurons that do something simple, which we can kind of understand. They kind of go to a certain firing rate and they keep it throughout the duration of the delay. But there are many other neurons that do kind of complicated dynamic things over time, and it's hard to tell how are these neurons contributing to this working memory. But the interesting fact that these guys showed is that if you look at the PCA projection of this population, actually the clusters which represent the different stimuli that the monkey has to remember maintain a stable representation of the stimulus over time. And this is kind of the interesting thing, something that's happening at the level of the population that otherwise we couldn't really infer from looking at the single neurons. Right, so let's get into some algorithms. So of course, you can't really talk about dimensionality reduction without mentioning PCA. Who here has never heard of PCA, completely not familiar with it? Right, exactly. So PCA is always a good starting point, even in neuroscience, to approach a data set. And what I would like to talk to you a bit about is also newer methods which target specific limitations of PCA that are of interest to us. So just to remind you what the data looks like, we have neurons recorded over time and across trials. So we can visualize this data, we can imagine this data as a third-order tensor, so this kind of block of data. We normally record action potentials, but we smooth them to obtain continuous time series, and the trials can be of different types and subtypes depending on the experiment. So PCA looks for a linear transformation which allows us to project our data into a lower dimensional subspace while either retaining as much of the variance in the original data or minimizing, and this is the formulation that I have here, minimizing a reconstruction error. When we do PCA neural data, there's many ways, but the most common one is that we say, okay, we have many trials, but probably the variability from trial to trial is just noise that we shouldn't really care about. So we can average these trials, basically compress our third-order tensor to a 2D matrix, and then we run PCA. But as you can imagine, there may be situations in which this averaging can be throwing away interesting things that we want to look at. And so a natural extension of PCA to 3-dimension is given by tensor component analysis, which is a recent rehashing of polyadic tensor decomposition. And essentially, it allows you to do PCA or some sort of decomposition on third-order tensors as opposed to second-order. And it looks similar. We again minimize a reconstruction error. There's a nice Python implementation that you can look up. And I will jump into an example to show you why this is interesting. So the crucial thing is that we can keep this third dimension that we otherwise would average out. And so we get information about the trials in our experiment. If you're not familiar with trials, normally the experiments are segmented into standardized repetitions, and this is what we call trials.
Sorry, to understand a bit, which is the dimension of these matrices? Are they big?
No, I mean, they are big for us. They're getting bigger. They're never big, so we record typically tens to hundreds of neurons. The time points can be relatively many, and we record, I would say, hundreds of trials. So it's still small data in any kind of larger sense. So to give you an example, this is data recorded by my ex-colleagues Jorrit Montaigne and Guido Meijer. What this experiment is, you have a mouse sitting in front of a screen, and on the screen we present a drifting grating. So the grating is essentially a sequence of alternating white and black bars, and it's drifting in the sense that it's moving this way or that way. And the reason for that is that we are recording in mouse V1, and we know the neurons in V1 are very sensitive to edges, to sharp changes in the image, and they have a preferred orientation. So every neuron will likely, so a lot of neurons will tend to prefer, for example, bars that are aligned like this, so they respond a lot whenever the grating is like this, and maybe a little bit when the grating is kind of vertical and not at all when it's horizontal. So they're selective to a certain orientation. And to give you an idea of what the data looks like in practice, we have these single unit responses. So this is time, and the shaded area is when we present the grating. And you see there are some neurons that do what I told you. They're selective for a certain orientation. So whenever the orientation is around 90 degrees, this neuron is responding a lot, and otherwise not so much. But there are also, and for example, similarly this one. But there are other neurons which, for example, depress their activity when they get an orientation that they don't like. Or other neurons that show an increase in activity which is kind of agnostic to the orientation. So we have a heterogeneous mix of responses. Yet PCA allows us to kind of compress all of this into some nice, this is just to show you kind of that this data, this is calcium imaging, is quite noisy as well. I don't know if you can see the individual trials. And when we run PCA on this data, we get quite a nice, simple representation of all of this mess. And so the first component in PCA captures a transient that is aspecific with respect to the type of orientation of our grating, and it just captures an increase in activity whenever the stimulus is presented. And later components capture smaller sources of variance which are given to the specific responses which are orientation tuned. And this is quite typical, that you have the first component capture the maximal variance is given just by the fact that whenever something happens on the screen, activity goes up, and then smaller differences in, for example, the type of trials appear in the later components. Oh, this is if you visualize these trajectories in 3D. And then I want to show you, so this is my blog, and there's a very long post on doing PCA on neural data with Python code to make all these plots, should you ever want to. And yeah, so this is what I wanted to show you. This is another visualization where you'll see every dot in this scatter is an individual trial over time. And you'll see that before the stimulus is presented, they're all kind of clustered in the middle. And then once the stimulus comes on, then these trials will naturally separate in PCA space and cluster based on the orientation of the grating. And this kind of looks something like this. And then when the stimulus is on, they separate. Of course, they're a bit noisy. And then when the stimulus comes off, they slowly go back to their resting state activity. And this is very simply done. This is all matplotlib and the animation module. And then you call this animation to HTML. And then, so these are, again, trajectories in 3D. And just for, because animation is nicer, animated trajectories where you see how trials separate based on this stimulus type that we present. So tensor component analysis is what I introduced before, where we do this decomposition directly on a third order tensor, which allows us to maintain this trial dimension that we would otherwise have to find a way to kind of squeeze with PCA. And the analysis that we get, the plot that we get looks something like this, where we have in this first column, we have the neural factors, is what people call them, which is essentially basically groups of neurons that show correlated patterns of firing. In the middle column, these are trial averaged typical patterns in activity which are expressed by the corresponding groups of cells. And then here we have the interesting addition, which is the trial factors, which are trial-specific amplitudes, which modulate these patterns. So essentially, in this specific trial, which is a blue dot, all these neurons are very strongly expressing this pattern, meaning that all the neurons that respond to that orientation are very active in that specific trial. And this is a completely unsupervised method, and it very naturally picks out that these neurons are orientation tuned, and we see that out of 5 components, 4 pick out a certain orientation and isolate a group of cells that do this behavior. So I hope you appreciate that this is in one plot, starting from like very messy. Of course, this is a system that we understand quite well, and orientation selectivity is relatively well known and very studied. But in one picture, we get a summary of kind of a lot of the things that are going on. And for example, we have this other component, which is activated only in 4 trials, which makes me suspect that this is a recording artifact. So in these 4 trials, something goes wrong. We have this noisy pattern, and this may be that something in our recording system is doing something wrong. And this is information that we otherwise would just compress and never find out that it was actually there, potentially. This is an example where we have more components, and again, this TCA method finds out more structure in our data. There's, of course, a whole topic which I'm ignoring, which is how do we find out how many components we need to describe our data, but I can't fit everything in this talk. So quickly, a second method is demixed principal component analysis, and the motivation for this is the following. So I told you that neurons are selective or respond to many task parameters. Many task parameters that we control, and probably a bunch of other things that we don't control. But specifically, if we have like the animals making different choices with respect to different stimuli in different locations, it may be that neurons are responding to all of these things at once. And it's kind of difficult to tease apart the effects that we're interested in. And if we run PCA naively, then the PCA components will preserve this mixed selectivity, meaning that they give us a valid representation of the data, very legitimate, but at the same time, it may be a little bit harder for us to interpret if all of these effects are combined in the PCA decomposition. So basically, demixed PCA was proposed as a way to essentially inform PCA of the task parameters that we care about in an attempt to separate subspaces which capture variance related to specific task parameters. So I have a compulsion to always put equations in my slides. I cannot avoid it, but you don't really have to understand it. This is just to show that this, coming from the minimization objective of PCA, this is just kind of, we decompose the data in a certain way, and we fit a bunch of kind of PCAs, if you will, but it looks essentially similar. So it's a kind of a deviation or an adaptation of PCA. And the idea is that this method kind of, it still tries to reconstruct. It's an in-between between what linear discriminant analysis would do and what PCA would do. So it tries to capture the geometrical relations of the data points like PCA and maximize the variance. At the same time, it knows about the different stimuli, so it can allow us to keep these subspaces apart. And I'll give you a quick example. This is another experiment in which, so a monkey sitting in front of a screen, and when a cue comes on, it knows what kind of action it will have to do. Then it has to wait a few minutes before, no, not a few minutes, sorry, a few seconds before it has to reach out and grab this object which is sticking out with 2 types of grip, either like a side grip or a precision grip. Right.
Excuse me, so
Yeah.
is the mixed PCA still applied to the temporal dimension? Or you average the PCA? You get
So no, it applies to the time dimension. So in this, so the thing where we still average over trials. So in that sense, it still averages over trials. So it loses the trial dimension, but it allows us to keep apart different task parameters of interest. So it kind of adds a supervision to PCA, but doesn't solve that problem. So if you look at what the single neurons are doing, again, heterogeneous mix of responses. You have, so the cue moment is where the monkey learns what movement it will have to do. The go signal, when it's told that it can start executed, and then an object touch is when it actually has reached the object. and then at reward, it gets a reward if it has done the right movement. So you can see there are some neurons which starts ramping up in activity before the monkey's even doing the movement. This is a recording in across motor and premotor cortex. So here already these neurons are starting to be active, and then here they do some kind of weird oscillation. Some neurons are active very specifically during the movement. Others are even depressing activity during this preparation time, and then doing again some sort of oscillation around the movement. When we run PCA on this data set, these are the first 9 components. Again, the green lines are the side grips, or one type of movement, and the purple is the other type of movement. You see that the first components, as in the previous data set, tend to capture changes in activity which are kind of agnostic to the child type. And then the differences between the two grips start to emerge in later components. But it's not exactly clear what these components are representing, because here we have again like a child type independent increase in activity, then differences between the child types. So I hope you appreciate that these components are kind of a little bit messy in a way. And oh yeah, so I show you here just some trajectories in 3D. Here the interesting thing is that even though the time between cue onset and movement start is very long, the distance in PCA space is very short, and actually between movement start and object touch, very short time, but the neural activity traverses a very long distance in PCA space. But so starting from this picture, basically DPCA allows us to say, hey, we're interested in time, we're interested in grip type, and we're interested in the force that the monkey is exerting. Can we get a cleaner decomposition? And yes, we can. So we have in the top row, we have time components. And this is components that are trying to capture variance over time, irrespective of differences in child type. Then we have the grip components, which are only focusing on the differences in grip. And then we have the force components. And this is I hope you can see that this is kind of a cleaner, although of course a slightly supervised picture of our data. What we can tell from this, for example, is that the monkey is not really, the activity of these neurons is actually not really separating between these two different movements that it has to execute before it actually has to do them. So one may think that maybe during this waiting time, the monkey is already preparing these two different movements, and then this preparation is represented somehow by the activity of these neurons. And as you can see, this is really not the case, and the two grips, the trajectories corresponding to the two different grips only separate at the moment where the monkey can actually move its hand and start the movement. And yeah, and then I show you again 3D trajectories. I just liked it because it looks like a butterfly. Yeah. Oh, yeah, and then to conclude, so this is, to summarize, dimensionality reduction in neuroscience. It's very useful to compactly represent our data, but in some cases it has, people argue that it can have a more profound meaning to identify these latent variables that span these low-dimensional manifolds. And although PCA is a good starting point to approach a new data set, there are newer methods that address some specific limitations of PCA in the context of neural data. And then I also, of course, this is all in, my work is all in Python. The two methods I showed you are implemented in Python. And I have a last slide because before I prepared this talk, I thought a bit about what makes me want to use Python as opposed to something else. And I guess specifically for the work that I do, neuroscience suffers from a lack of standardization, meaning that every data set that you'll get will be structured in a completely different way, and the information is all there, but it's kind of, every neuroscientist has its own way of managing and preparing data. So you always have to do, I always have to do a lot of operations before I can get to the point where I can call scikit-learn or I can call TCA or all these things. And I think this is where Python really makes a difference for me in the flexibility and the ease of use of its data structure, which allow me to kind of do all these annoying manipulations very quickly. Also, our analysis is very exploratory. So it's hard to set up a pipeline where you can just feed in new data and expect it to work, because every data set will have its quirks and its differences. And so in that sense, having a language that allows you to script quickly in a way that remains readable also over time is a great advantage. And with this, I think I finished. So thank you very much.
Questions?
Hi, thanks for the nice talk. I was wondering if you do embeddings in more complicated manifolds, because PCA, if I assume correctly, there's just linear space subspaces. Yeah. So are there any methods you use there, just out of curiosity, which are more complicated in a manifold sense?
So you have this whole kind of field of manifold learning, of course, which does nonlinear dimensionality reduction. In my experience, they work well on toy examples, and then when you try to run them on real data, it's always a bit too noisy, and then you get a mess out of it. And to further elaborate on this, the hope is that we can do linear dimensionality reduction, because if our data lives on this sheet, which is a nonlinear, which is a manifold, but it's not linear, then hopefully for the kind of region of neural space that we explore during a certain task, we can approximate the manifold locally linear with a plane in 2D. So hopefully we can keep doing linear dimensionality reduction, because I don't think that algorithms are as robust and as good to do nonlinear.
Thank you.
Thank you for your talk. In addition to dimensionality reduction techniques, do you also use clustering techniques to understand, let's say, group all different neurons that behave in the same way, or something like that?
Absolutely, yeah, absolutely. So if you, clustering techniques, or sometimes you do dimensionality reduction and then clustering on PCA, yeah. I personally have done it a few times. I don't always do it, but definitely, yeah.
Okay, thank you.
I mostly what I do is also decoding. So basically, you try to train machine learning models to predict certain task parameters from the neural activity. That's a very common. Yeah.
Any more questions? Thank you.
All right, cheers.
Our next talk will be navigating the magical data visualization forest.
So great, thank you for listening to my talk. I want to talk about data visualization, and I came up with this title basically because I've done a PhD and looking a lot of forest data, and now I went into working for IBM, doing data science and developer advocacy, and this talk is kind of trying to use my forest data from my PhD, doing what I do now. And this better? Yeah. Great. So I'm just going to jump in because I only have 30 minutes.
It's much worse, actually.
Is it worse? Better? Yeah. So I'm going to go quick because I actually have only 15 minutes, not 30. All the slides and notebook that I'm using are on GitHub, and this is an experiment for me because I made the slides in a notebook. So if anyone's really good at it and knows how to make them a little bit more interesting, I would love to talk to you. That's as an aside, because this is how far I got with the layout and things like that. And so yes, I'm talking about visualizing data, and especially in Jupyter notebooks. And of course, probably everyone in the room here does this already. Curious to know who has not visualized any data in a Jupyter notebook. Exactly, nobody. So I'm guessing most of you doing this locally, so using your Linux, Windows, Mac, using Anaconda installation or whatever. But as I am working mainly for the IBM cloud, I am very interested also in looking into using Jupyter notebooks in the cloud, because that gives a bit of a different way, because your data comes from a different way. It's stored somewhere in a database normally, or an object store, and really everyone offers these kind of notebooks in the cloud now. Amazon, Google, IBM, Microsoft, many others. I haven't really made a full, complete list here. And so back to visualizing data with Python. There's many packages to do this. We've heard quite a few over the last days here. Yesterday, a really interesting workshop about matplotlib, I thought, and just earlier about Altair. And originally in my talk, I wanted to give you a few examples of how that works, but I'm sure you all know how that goes. So I would say it is a lot of different packages, and they're all slightly different. So the thing I have to do all the time is look up the syntax, because I just can't remember, especially if I switch between different packages. So I really enjoy doing all this data visualization. I spent hours, and I probably wasted or spent months in my PhD on making the perfect figure to show exactly what I wanted it to say. But in reality, if you're just for the first time looking at your data and you're very interested in how do I do this, what's actually in there, I think there is a quick way, and that's what I wanted to talk about. An easy way, which is basically pretty much a wrapper or open source package, Pixie Dust, that uses all the other different, not all of them yet, but it potentially could use all the different visualization packages and turns it in a simple wrapper. And it's Pixie Dust. I've been working on this now for 2 years, because in my team, in developer advocacy, we're really interested in helping people out, getting into open source, also help people get started with coding. So a lot of people, oh, well, I use Excel, I will never be able to code. So how do you make it a little bit simpler for people like that? And then that's how this project started. And it kind of explode in every single direction from there. So it's really simple. You install it through pip install Pixie Dust. It's on PyPy. And big question, of course, is what does it do? It does a lot of things. It started off because we were looking at how do we get people, convince them to work in the cloud. That was the whole idea, of course, but it is open source, and I think it's actually super useful anyway. If you're not if you're working with Jupyter notebooks, so how do you install kernels? So, for instance, the Spark kernel on my local machine, it helps you through the steps of doing that. Loading data, obviously it's not that hard, but it's included. Then the main thing I want to talk about, and I will talk about in a bit of a demo later, how to display data using all the different packages, and also filtering data. And the things, unfortunately, I have no time for, but I think they're very cool, is you can also build interactive data visualizations, very similar to our Shiny apps in R, obviously. And then you're using interactive files where you can click and add buttons and have dashboards in different tabs and make it a big dashboard inside your notebook. And then when you've built such a thing, I think it's nice that you also have now the option to publish these apps straight away through a Kubernetes cluster and as a web service, which you then can embed in any other page or wherever you're looking at. And the last thing, if I have time, I'll show you. We also added a debugger to debug your code in a notebook, and I have to say the names of these. So these are called Pixie Dust is the package. Then we have Pixie Apps, the Pixie Gateway, and the Pixie Debugger, and they're all like in a big family that seems to grow, and things that we think are useful for notebooks are added. So let's go to the notebooks. And is this big enough? So as I said before, it's very simple. I load in my package with import pixie dust, and I might sit. And then there's a few things that I wanted to show you. So the first thing is there's now pixie dust sample data, which gives you just a bit of data to play around with. But then you can load this data, but just by adding the number to it. But much more interesting, of course, especially when you're working in the cloud. I think Pandas has this as well. But in this case, you can just say pixie dust, load my data, and give it a URL. And then you have the option to either import it as a Spark data frame or as a Pandas data frame. And if you want Pandas, you need to add force Pandas true. And as we started off with Spark, and then we realized people want actually Pandas, that's how it ended up this way, really. And then I'm just going to show you a little bit more. So I loaded this data, which is the CO2 concentration over the last 40 years in Mauna Loa. And in this case, I'm just loading it locally, because obviously that's a lot safer in a live demo. And this is where Pixie Dust really comes in. I'm going to run this cell now, saying display my Pandas data frame, and then it comes up with this table. You can look at the schema, you know which years, which variables are in the table, what the types are, and get rid of this, and look at the table itself, and that's, because the screen is so low, normally this is on the side, but it's now at the bottom, so you can hide all these things as well. And then the cool thing here is there's also a filter option. So if you have new data, you have no idea what you're looking at. I'm just going to have a look around at my data, and I want to only look at when the CO2 concentration is above 400 ppm. I'm going to say apply, and then your table is updated. And this filter function will work on whatever you do after this. So what I'm going to do now is go to the next cell. I had quite a lot of missing values, so I'm taking these out first. Then I'm going to run that cell. And then instead of going to Matplotlib or any of the other ones, I'm just going to, I know what kind of data I have, and now I can just click inside the notebook. I want to make a line chart. And I've done this already, so that's the cool thing. It does remember in the background what you have done. But if it's the first time, I have to make it smaller because I can't. It's putting things next to each other. It's easier. So I put this is the time in decimals and the ppm, and I'm going to make a plot. In this case, this is the raw data. Here I'm using bokeh, but then I can use matplotlib, or I could use Brunel, and these kind of things. So there's a lot of things you can do, and I also can cluster by any other kind of thing. I can do many other charts. I can do, now got stuck. So I can do bar charts, and cluster them by different numbers. And there's also seaborn, so here I did a scatter plot on forest data, which is quite a bit different. And I did the same kind of data as well as a map in Brunel in this case. And there's also the Pixie debugger. I'm just going to show it quickly, which is great because this is basically how I write this package. The only thing you have to do is add a bit of magic, Pixie debugger, and then when you put that in a file, you can jump through this through the code and find your error. And if you have an error like this, you just the next file you say just 1% pixie dust, and when that will run, it should come up. So this is basically the package I wanted to talk about you quickly, show you what it is. And as we are now kind of developing this guide, I'm really curious to hear what you think about this kind of package. Would you use it? Would you think it's useful? If you're super interested in creating issues or adding your own data visualization. And the other thing I want to mention, there's a book out. I have reviewed it, and David, who worked on it mainly, he went through how to exactly build all the Pixie apps and make it interactive and set up on a Kubernetes cluster. And that's pretty much it. And this is what I want you to remember. Import Pixie Dust and then display. And I think the best way is just get your own data and play around with it. Thank you. And check out this link.
Thank you for your talk. Are you thinking of contributing upstream the debugger to Jupyter, or would that be possible?
Yeah, I think David is already talking to them, but he's in the US, so we don't speak that much.
Thank you. Hey, so just looking at your presentation, I was wondering, I found it considerably more confusing to navigate through the entire drop-downs and switch views than the entire attribute structure of the packages is. I understand you're maybe trying to go into the niche of people who are not familiar or comfortable with the command line, but have you looked at in how far that's really something they would be more amenable to? Because it's more graphical, but I don't know in how far it's less complex.
Well, I find it easier because of just quickly exploring your data is a lot quicker. But if you want something in, of course, publish ready, you have to go back to matplotlib and make it right.
Any more questions? Thank you.
Thank you.
Talk will be 3 most common mistakes in data visualization.
Can you hear me? Yes, excellent. So let's talk about data visualization. It will be the first time today, right? I will talk to you about the three most common mistakes in data visualization and how to avoid them. And so if there is one thing that I want you to remember from this talk, is most probably you're doing data visualization wrong. Most probably, I'm not. So my name is Boris. I'm a data scientist at Automatic. Automatic is the company that produces WordPress.com, WooCommerce, Jetpack, and other cool projects. I also teach data visualization in 2 colleges in Israel. And for me, data visualization is not only a tool, it's just also a hobby and a passion. Automatic is a very nice company in many ways. One of those ways is that we are a distributed company, so it doesn't matter where you live. As long as you have a computer and a network connection, you can work at Automatic. So we are constantly looking for new employees, and I highly recommend working at this company. So let's go back to data visualization. Why do we use data visualization? And it turns out that we are not that aware about the fact that there are 2 reasons to perform data visualization. And those reasons are not overlapping. So the overlap isn't complete. So we do data visualization to explore things like we saw in the previous lecture, but we also do data visualization to explain things, to explain concepts. Okay? And explaining is not exploring, because exploring, I explore new things in order to learn new concepts or to learn solutions to problems, but I explain things in order to teach something. And I'm not talking only about teaching in an academic institution, but also, let's say I have a model and I want to convince you that my model is good and you need to adopt my point of view. It's also teaching, it's convincing. And convincing is not the same as understanding, because I can understand something, and I understand the problem, and then understand the solution, but I will not convince you about it, right? And convincing is a very hard problem. There are 2 different problems. Exploring is a very complicated thing, and all you have to do is to learn, to have a good tool and to learn how to use it. But to explain things, you also need to know how the human beings work, right? Because you have to know how people perceive colors. You have to know concepts about physiology of vision and psychology and many other things. That's why I am fascinating about this part of data visualization. And just to show you some examples about how crucial it is, take a look at this study that was performed in 2011. In this study, the researchers tested 2 different color encodings for a diagnostic machine that diagnoses a certain heart condition. So a physician looks at the screen and decides whether this person has a heart disease or not. And it is a very serious problem, right? And the decision, a wrong decision has a very high cost. It turns out that by changing the color scheme alone, they were able to improve the rate of correct diagnosis from 62 to 91%, only by changing the color scheme. Okay, so it's not only about it's a matter of taste. It can be even a matter of life and death. And in a more relaxed way, take a look at this pair of graphs. Those graphs are depicting a gender payment gap in the United States. The data in those graphs are completely the same. But people who look at this graph tend to think that the gap is smaller than people who look at this graph, only by changing the order of the pink and blue bars. So if I'm talking about convincing, now when I want to convince you that the gap isn't that big, I will show you this graph. And if I want to convince you that the gap is bigger, I will show you this graph. The data is absolutely the same. Okay? So it's not only about you as a presenter who presents something and try to teach. It's only about you as an audience who look at graphs and try to learn. So keep in mind that somebody can and do manipulate you by just using those tricks. So if you don't know those tricks, somebody does and will try to trick you. And there are dozens of studies performed every year in field of data visualization, starting from the early 1930s. So the first out of the three misconceptions about data visualization is that actually there are two tasks, explanatory and exploratory. And also we are not, many people are not aware that data visualization is an evidence-based field of applied science, if you will. Okay? So it's not only about a matter of taste or nice colors, there is actual science in it. And this is the attitude that I want you to keep in mind next time you create a presentation slide. Let's go to the second misconception, the second error. Some people claim that we have an attention span that is shorter than that of a goldfish. Some people claim that it is not shorter than that of a goldfish. But goldfish or not, we will all agree that our attention span is limited, right? Which is a problem, because we are constantly flooded by information, and you as a presenter want to provide to give as many information as you want, as you can. But there is a lot of information that is streaming into our minds. It is my presentation, my voice, your own problems. That is why there is a concept that is called data-ink ratio that is very close to the signal-to-noise ratio that is familiar to any physicist or most of the scientists. And this concept says that if you take a look at a graph, some graphical elements on this graph represent actual data, and other graphical elements on the graph don't represent any data at all. And according to Professor Tafti, the rate between the data ink and the non-data ink has to be as big as possible. In other words, we need to maximize the signal-to-noise ratio, which means that if your graph doesn't have any data, it should be blank. No data, no ink. And the problem is that we all agree with this, but most of us produce graphs that are rich of unused data. Excessive grid lines, excessive colors, this is all useless data. Okay? So we need to remember this. We need to keep this signal as high as possible and the noise as low as possible, or data to noise. But keep in mind that data and signal has to be useful. So we need to maximize the useful information and minimize the useless information. And usefulness is not an objective thing, because different people, different audiences have different concept about usefulness. I think that most of us will look at this graph and will not understand anything. Well, we know that this is a ECG, right? But unless you are a trained physician or ECG technician, you don't know to read this graph, despite the fact that all the information is here. The same goes with other graphs that we see and produce. So box plots are very useful for many scientists, but many people find them very, very confusing. And sometimes we should simplify them. And maybe a social scientist will find more value in this type of graph that only depicts the differences between the payments. Okay? So you need to select the form of the graph that you provide and the composition of this graph. And this is the second problem. Many graphs are rich of useless information, and many of them use inappropriate graph types. I mean, sometimes a graph type is appropriate for you, but not for your audience. And when you switch the audience, you should reconsider the type of the graph that you are using. And this is the third problem, text. You see, when you learn to read, you cannot unlearn it. When you see some text, you will read it. So when you see a slide with text, everybody will start reading those slides, even if they don't want it to read it. Okay? The problem is that many slides contain many conflicting text labels. So we have predicted versus observed here, and observed versus predicted here. And the conflict is pretty obvious, okay? So we need to remove the conflicting titles and to keep only one title, to keep the signal-to-noise ratio up. But this is not the only problem. This title does not add any value to this graph, because most of you already know that this graph depicts predicted and observed values in their agreement, which means it doesn't produce any useful information. What I claim is that in most of the cases, when you are done building a graph, you should add a title that says not the what the graph shows, but the so what, the conclusion. And you gain two things, or maybe three things with this. First of all, you discipline yourself. Once you force yourself to formalize the conclusion and put the conclusion on the graph, you force yourself to review the graph and ask yourself, really? Does it mean what I wanted? Does it say what I wanted to say? Maybe not. Maybe I should improve this graph. Secondly, it tricks your audience to accept your point of view, which is a cynical thing, but it works. I mean, it works. So if you don't want to be very bold, and you can be a little bit more cautious about yourself, but then again, you have to take risks sometimes, because when you study something, you come to a conclusion. You have to take to stand behind those conclusions. So my third point is, don't title your graphs with the what, but with the so what. And it also means is valid about captions in figures and texts. So instead of saying observed versus predicted, I claim maybe you should say figure 1, good agreement between model and the predictions. And some people think that it is a very cynical and unacademic way of work, of action. I don't know. I wrote about it in my blog, and if you think, I'm very open about somebody to change my mind. I think it's completely okay. So if you think I'm wrong, go to my blog, look for C-form conclusion, and argue with me. I'm open for that. So those are the three most common mistakes. Bad attitude. Remember that data visualization there are actually two things, explanatory and exploratory, and it is very much evidence-based. Graph contents and composition. Keep your graphs clean, and graph types, feed the graph types to the audience, and write your conclusion. Okay, any questions? Yes, thank you.
I liked your presentation a lot. Very many people seem to want, and more recently some people seem to want to produce interactive visualizations, like visualizations where you go on a website and you pull on a slider allegedly in order to understand the data better. It was always my view that interactive visualization actually meshes up the learning and the teaching of data visualization in a very poor way, where it doesn't train people to basically summarize the data in an efficient fashion, and it trains them to explore it in a very disorganized way, which is very bad for then communicating findings. What do you think about interactive data visualizations?
Interactivity for interactivity is a bad thing, but it is the tool. And as any tool, it can be used very efficiently. So I suppose you are familiar with the presentation by Hans Rosling, the late Hans Rosling, about the change in life expectancy and income over the years in the entire world. And it is an interactive, animated presentation which was fabulous. So as any tool, it can be used and it can be abused. So it's a balance. You have to decide whether it's a useful use or useless use of.
Slightly cheeky question, but the previous, the presentation in the other room finished very late, so I think a bunch of us missed your presentation. Would you mind just giving a 5-minute recap of what you said when everyone else finishes asking their questions? That would be amazing.
Thank you for your question. I have a slide for that.
Okay.
So, because the title is 3 mistakes and how to avoid them. So on the way to Trento, I had like epiphany. I have devised a formula, which I call a simple to use, easy to follow, never failing formula for data visualization. And I call it the ABCD of data visualization, which works like this. When you start producing a graph, you first have to think about your audience, who your audience is. Is it your colleagues or general public, or I don't know. Then you build your graph. Then you write the conclusion, and then you delete everything that is unneeded for your conclusion. Thank you. And there's the YouTube of the tutorial.
Any more questions? Thank you.
Thank you.